Welcome to the Medicine Podcast Zone. Today, we will have Genetic Biochemistry and Hereditary Disorders, Part 1.
That is quite the mouthful of a title to kick us off.
It really is, yeah. But I mean, usually when we approach a medical diagnosis, there's a certain expectation of mechanical precision, you know?
Right, like a visible cause and effect.
Exactly. Like you break a femur, the radiograph shows a jagged fracture line, and the orthopedic surgeon just points to it and says, there is the pathology. It is incredibly comforting because the defect is visible.
Yeah, it's macro and it's neatly categorized. It's totally binary. The bone is intact or it's fractured. The artery is patent or it's occluded.
Right. But genetic biochemistry just strips away that macroscopic comfort entirely.
Oh, absolutely. We are operating in a diagnostic landscape where the pathology is just invisible to the naked eye. It's written into the very architecture of the cells.
So we are diving into the incredibly complex blueprints of human life today. And the mission for this deep dive is to demystify the mechanics of inheritance for you.
We want to unpack how our genes are passed down right and how those specific alleles shift across massive populations over time.
Yeah, and we'll get into the highly specific, sometimes frankly bizarre, mechanisms behind hereditary disorders. Today's sources are some really dense medical notes and diagrams covering genetic biochemistry.
Extremely dense. But our goal is to extract the most fascinating nuggets. We're pulling apart the underlying math of human evolution and the specific quirks of genetic disease without, making you feel totally overwhelmed.
What's truly fascinating as we navigate this clinical landscape is the duality of the human code.
Yeah, we are looking at profound resilience side by side with terrifying fragility.
It really is the story of how a single microscopic typo-like one-swapped base pair among billions can cascade into a systemic life-altering condition.
I know, it's wild to think about.
I'll make sure we anchor this with clear analogies so we don't get lost in the molecular weeds. But I'm counting on you to provide the rigorous why behind the pathophysiology.
I can definitely do that.
Okay, so before we even consider how diseases travel through family trees, we have to establish the baseline of how genes misbehave on an individual level.
Right, because there are specific glitches in the code that completely defy straightforward Mendelian logic.
Let's start by examining dominant negative mutations.
Yeah, this phenomenon completely upends the classic high school biology concept of recessive and dominant traits.
How so?
Well, usually if a patient has one functional allele and one mutated allele, haploinsufficiency might be an issue, or the functional copy might just upregulate to compensate.
Right, the good gene works a little harder.
Exactly. But a dominant negative mutation exerts a dominant effect in a deeply destructive way. The mutated allele produces an altered protein that actively antagonizes the wild-type protein produced by the healthy allele.
Think of this like a high-stakes group project. A standard recessive mutation is the team member who simply fails to show up.
Yeah, slacker.
Right. The rest of the team works twice as hard, compensates for the absence, and you still deliver the project.
Which is how most carrier states work.
Exactly.
Yeah.
But a dominant negative mutation is the team member who logs into the shared server and actively deletes the functional code written by the rest of the team.
They literally sabotage the entire system.
Yes. They don't just sit out, they ruin it for the functioning members.
That is precisely the molecular mechanism. A really high-stakes clinical example of this is the p53 tumor suppressor gene.
Oh, the guardian of the genome.
That's the one. The p53 protein functions as a tetramer, meaning it requires 4 individual p53 subunits to bind together to effectively attach to DNA.
And its job is to halt the cell cycle when DNA damage is detected, right?
Exactly. But if you have a single mutated p53 allele, you are producing malformed subunits.
And because those malformed subunits still bind with the healthy wild-type subunits, they poison the entire tetramer complex.
You've got it. The mutated protein physically occupies the space, forming a dysfunctional complex that cannot bind to the DNA promoter.
So the wild-type p53 is essentially handcuffed to a defective partner.
Yes, rendering the cell entirely incapable of suppressing tumor growth. The mutated protein actively blocks the rescue operation.
Wow. Okay, so moving from the behavior of a single mutated protein to how genes arrange themselves on the chromosome, we encounter linkage disequilibrium.
Which, frankly, sounds like a statistical nightmare.
It really does. Break it down for us.
The terminology is dense, but the concept is anchored in physical proximity. Linkage disequilibrium is the tendency for certain alleles at two linked loci to occur together more or less often than expected by chance.
And it's crucial to remember this is a population-level metric, right? Not a family pedigree metric.
Exactly. We measure this across entire populations.
Because normally during meiosis, homologous chromosomes cross over and swap genetic material. It's the great genetic shuffle that ensures diversity.
Right, which makes you wonder, why wouldn't two alleles randomly separate over thousands of generations?
Exactly. Why do some always stick together?
Because they are physically located too close to one another on the chromosome strand.
Oh, so they're literally neighbors.
Yes. The physical distance between them is so small that a recombination event is statistically unlikely to occur precisely between them. So they are inherited as a contiguous block.
What are some examples of that?
The HLA loci on chromosome 6 or the CFTR gene variations are prime examples. We see specific HLA haplotypes dominating certain geographic populations because those gene clusters have traveled together unbroken for millennia.
So they're geographically tied together on the DNA map, meaning they just commute through generations together.
That's a great way to put it.
But what happens when the genetic map itself is inconsistent within a single human body? That brings us to mosaicism.
Ah, yes. The presence of genetically distinct cell lines in the exact same individual.
Which is wild to think about. How does that happen?
Well, mosaicism depends entirely on the timing of the mutational event.
Okay, timing is key.
Right. If a mitotic error occurs after fertilization, so after the zygote has already begun to divide, we see somatic mosaicism.
Meaning a mutation occurs in one specific cell during embryogenesis.
Yes, and as that cell proliferates, it creates a distinct lineage of mutated tissue. Meanwhile, the rest of the body develops from the unmutated cells.
So the patient literally becomes a genetic patchwork. Some tissues carry a severe mutation, while adjacent tissues are completely wild type.
Exactly. Contrast that with germline, or Gonadal, mosaicism.
How is that different?
In this scenario, the somatic cells of the individual are wild type, perfectly normal, but a mutation occurred specifically in the lineage of cells destined to become gametes.
Wait, so their body's fine, but their eggs or sperm carry the mutation?
Yes, which presents a terrifying diagnostic trap for genetic counselors.
I can imagine. You have parents who are phenotypically completely normal. They test negative for mutation in their blood.
But yet they have a child with a severe autosomal dominant disorder.
And the assumption might be a de novo mutation in the child, just a random new error.
Sure, but if they have a second child with the exact same disorder, you have to suspect germline mosaicism. The parent's reproductive cells harbor a hidden defect.
That's so tricky.
It is. Now, to illustrate the extreme physiological stakes of somatic mosaicism, look at McCune-Albright syndrome.
Okay, let's look at that.
The pathology here is an activating mutation in the GNAS gene, which encodes the alpha subunit of a GS protein.
And a GS protein activates adenyl cyclase, creating a massive influx of cyclic AMP, right? Which acts as a cellular go signal for proliferation and hormone production.
Precisely. So if that GS protein is locked in the on position, the downstream cascade is catastrophic.
What does that look like clinically?
It presents with unilateral café au lait spots, specifically with these ragged coast of mane borders.
Interesting description.
Along with polyostotic fibrous dysplasia, where normal bone is basically replaced by proliferating fibroblasts and collagen.
Oh, wow. And does it affect hormones too?
Yes, it triggers severe endocrinopathies like precocious puberty because the endocrine glands are just autonomously hyperfunctioning.
But the truly mind-blowing aspect of McCune-Albright is that a germline or prezygotic mutation of GNAS is fundamentally incompatible with life, isn't it?
Absolutely. If every single cell in a developing embryo had an actively locked-on GS protein, the embryo would arrest and die almost immediately.
The profound hyperproliferation would destroy any chance of organized organogenesis.
Right. The patient only survives because the mutation occurs after fertilization. It is a somatic mosaicism.
So the disease only affects the fraction of their tissues derived from that single mutated precursor cell.
Yes. It is literally a localized, survival-enabling glitch.
Timing really is everything. It dictates the difference between embryonic lethality and a viable, albeit complex, clinical syndrome.
Exactly. Now, we also have to consider how completely different genetic errors can result in the exact same clinical presentation.
Right, let's unpack locus heterogeneity versus allelic heterogeneity.
This is a crucial concept for understanding why genetic screening panels have to be so exhaustive. Locus heterogeneity means mutations at completely different genetic loci like entirely different chromosomes can produce the exact same phenotype.
Give us an example of that.
Albinism is a perfect example. You could have a defect in the tyrosinase gene on chromosome 11, or a defect in the OCA2 gene on chromosome 15.
Because the biochemical pathway of melanin production requires multiple sequential steps.
Exactly. Knocking out any of the different enzymes along that assembly line halts production, resulting in the exact same clinical albinism.
Okay, so the destination is identical, even though the roadblock is on a different highway.
That's a great analogy. Now, allelic heterogeneity is the inverse. You have different mutations, but they all occur at the exact same locus.
And beta thalassemia is the classic presentation for this, right?
Yes. There isn't just one beta thalassemia mutation. There are hundreds of distinct point mutations, small deletions, or promoter defects that can occur within the beta-globin gene on chromosome 11.
And depending on whether that specific allelic mutation causes a total absence of the beta-globin chain or merely a reduced production, the patient's clinical severity will vary wildly.
Right, ranging from a mild microcytic anemia to transfusion-dependent thalassemia major.
It really exposes the limitations of overly simplistic genetic testing. You can't just assay for a single base pair swap.
Not at all. Now, speaking of varied clinical severity, we need to address heteroplasmy and the unique rules of the mitochondrial genome.
Yes, the mitochondria, the powerhouse of the cell, as everyone remembers.
And mitochondrial inheritance operates outside the nuclear genome entirely. Mitochondrial DNA, or mtDNA, is a small, circular chromosome inherited exclusively from the maternal lineage.
Because the oocyte provides the mitochondria for the zygote, and the sperm's mitochondria are essentially destroyed upon fertilization.
Right. But a single eukaryotic cell contains hundreds of mitochondria, and each mitochondrion contains multiple copies of mtDNA.
So heteroplasmy is the coexistence of both normal and mutated mtDNA within a single cell.
Exactly. And during mitosis, those mitochondria segregate randomly into the daughter cells.
It's just a localized numbers game then.
It really is. By pure chance, one tissue lineage might inherit a massive burden of mutated mitochondria, while another tissue lineage gets mostly wild type.
Which creates a threshold effect. A tissue won't show symptoms until the proportion of mutated mitochondria crosses a critical functional threshold.
Which explains the massive phenotypic variation we see in conditions like Leber hereditary optic neuropathy.
How does that play out in a family?
You could have 3 siblings inheriting the exact same maternal mtDNA mutation. One sibling might suffer subacute bilateral vision loss because their retinal ganglion cells received a high burden of mutated mitochondria.
While another sibling remains completely asymptomatic.
Exactly. The systemic distribution of the mutation is just chaotic.
Wow. Okay, the final anomaly of individual inheritance we must dissect is uniparental disomy, or UPD.
Yes. Normally, you inherit one homologous chromosome from your mother and one from your father.
But in UPD, the offspring inherits 2 copies of a chromosome from a single parent and 0 copies from the other.
Let's trace the mechanics of this because it requires multiple massive errors during cell division.
Okay, let's break it down.
We have heterodisomy, which indicates an error in meiosis first. The homologous chromosomes fail to separate, so the gamete ends up with both the grandfather's and the grandmother's version of that chromosome from one parent.
And then we have Isodisomy, which is an error in meiosis second, where sister chromatids fail to separate.
Right. The gamete gets 2 perfectly identical copies of a single chromosome, or isodisomy can occur postzygotically.
Meaning a trisomy might be rescued by the random loss of one chromosome, leaving behind 2 identical copies from one parent.
Yes, exactly. But here is the critical physiological reality of UPD. The individual is euploid. They have exactly 46 chromosomes.
So they have the precise correct dosage of genetic material.
Right.
But if the dosage is entirely correct and the gross chromosomal architecture is normal, how does this present a pathology?
In many cases, it doesn't. A patient could live their entire life entirely unaware they have UPD for certain chromosomes. But isodisomy creates a profound diagnostic trapdoor. Imagine a scenario where a mother is a heterozygous carrier for a rare autosomal recessive disorder. The father is completely homozygous wild type.
According to standard Mendelian Punnett squares, it is statistically impossible for them to have an affected child. The child could only ever be a carrier.
Correct. But if an isotomy event occurs and the child inherits 2 identical copies of the mother's mutant chromosome and loses the father's healthy chromosome,
the child suddenly has a double dose of the mutated allele, They manifest the severe recessive disease despite only having one carrier parent.
It completely shatters the expected mathematical probability of inheritance.
It also deeply impacts imprinted genes, which rely heavily on having both a maternal and paternal copy present to function properly.
Which brings us to a critical transition. We have looked deeply at how alleles mutate, misfold, and missegregate in an individual cell.
But what happens when we zoom out from a single family pedigree? When we examine how these alleles propagate across millions of individuals over thousands of years,
We have to understand population genetics.
yes, we are shifting from the cellular microscope to the evolutionary telescope. How does the human code shift over time?
There are 4 primary engines that drive shifting allele frequencies in a population.
Let's start with the most dramatic, the bottleneck effect.
The bottleneck effect is a radical, non-selective reduction in population size. Imagine a highly diverse population where multiple alleles confer roughly equal fitness.
Okay, a balanced ecosystem.
Then a catastrophic event occurs. A massive volcanic eruption, a famine, or a lethal pathogen. This event wipes out 90% of the population indiscriminately.
So the survival of individuals isn't based on their genetic superiority at all. It's based on sheer luck.
Precisely. The people who happen to be on the other side of the mountain survived. The genetic makeup of those survivors becomes the absolute baseline for the new rebounding population.
Entire alleles can be eradicated from the gene pool simply because the individuals carrying them were in the wrong place at the wrong time.
Yes. Now, the founder effect operates on a very similar principle, but instead of a mass extinction, it's caused by a calamitous population separation.
A small splinter group migrates away from the main population and founds a geographically isolated colony.
And whatever specific alleles those few founders happen to carry will dominate the new population.
So if one of the original 10 founders happened to be a carrier for a rare recessive disease, the frequency of that disease in the new isolated colony will eventually skyrocket compared to the broader global population.
Exactly. Both the bottleneck and founder effects are extreme examples of genetic drift.
Genetic drift being the random statistical fluctuation of allele frequencies from generation to generation, independent of any survival advantage. It is literally the universe simply rolling the dice.
Contrast that with the fourth force, natural selection. This is not random.
Right. Natural selection is the directional pressure exerted by the environment.
Alleles that confer a physiological advantage, be it better metabolic efficiency, resistance to a prevalent pathogen, or enhanced reproductive success, will systematically increase in frequency.
Because the individuals carrying them are just more likely to survive and reproduce.
Exactly.
But how do we actually measure these forces? How do we prove that a population is actively evolving?
This requires the bedrock mathematics of population genetics, the Hardy-Weinberg principle.
Okay, let's get into the math. The Hardy-Weinberg equilibrium provides a mathematical framework to calculate expected allele and genotype frequencies.
Right. If we define P as the frequency of the dominant wild-type allele and Q as the frequency of the recessive mutant allele, the sum of those frequencies must equal 100%. So P plus Q equals 1.
And to calculate the distribution of those alleles into actual human genotypes, we square the equation. The resulting formula is P squared plus 2PQ plus Q squared equals 1.
Let's break those variables down for everyone.
Okay, P squared represents the homozygous dominant individuals. Q squared represents the homozygous recessive individuals, the people who actually manifest the disease.
And 2PQ represents the heterozygous carriers.
Got it. What about X-linked diseases?
For an X-linked recessive disease, the math changes slightly because males are homozygous. They only have one X chromosome. Therefore, the frequency of the disease in males is simply the allele frequency, Q.
And for females to manifest an X-linked recessive disease, they need 2 copies, so their frequency is Q squared.
Precisely.
Yeah.
But the Hardy-Weinberg equilibrium rests on an incredibly fragile set of theoretical assumptions.
Right. For the allele frequencies to remain perfectly constant across generations, there could be no new mutations occurring at the locus.
There can be no natural selection.
Mating must be perfectly random.
There can be no net migration or gene flow into or out of the population.
And the population size must be infinitely large to negate genetic drift.
None of which actually exists in the real world.
Exactly. Perfectly random mating, zero migration. It is a biological impossibility.
It truly is.
So if the foundational assumptions of the equation are functionally impossible, what is the clinical utility of the math?
That is the crucial conceptual leap. Hardy-Weinberg is not a description of reality. It is an idealized null hypothesis. It establishes a perfectly stagnant baseline.
Oh, I see.
When we measure the real-world allele frequencies of a population and find that they deviate significantly from the Hardy-Weinberg predictions, that deviation is the mathematical proof that evolutionary forces are actively at work.
It allows us to quantify the exact rate at which a population is migrating, mutating, or undergoing natural selection.
It is a yardstick to measure the velocity of genetic change.
Let's apply this clinically to cystic fibrosis in the United States, because the math reveals an incredibly hidden reality here.
Let's do it. Cystic fibrosis is an autosomal recessive disease, and the epidemiological data shows a prevalence of roughly 1 in 3,200 live births in populations of European descent.
In the framework of Hardy-Weinberg, those affected individuals are represented by Q squared. So Q squared equals 1 over 3,200.
To find Q, the frequency of the mutant allele itself within the broader gene pool, we take the square root, which gives us approximately 0.017 or 1.7%.
And since P plus Q equals 1P, the frequency of the healthy allele is 0.983.
However, a highly useful clinical shortcut is that for rare diseases, we can approximate P as being essentially equal to 1. The math barely shifts.
Okay, so using P is roughly 1 and Q is 0.017, we want to calculate the carrier frequency, the people who harbor the mutation but have no symptoms.
Right, that is the 2PQ term.
2 multiplied by 1 multiplied by 0.017 equals 0.034. That is 3.4%, or roughly 1 in 28 individuals.
Stop and process that statistic for a second.
1 in 3,200 people actually suffer from this devastating disease, but roughly 3.4% of the general population are silently carrying the mutant allele.
Completely unaware, perfectly healthy, passing it down through generations.
It perfectly illustrates how an allele can be incredibly common in a population, even if the clinical disease is relatively rare.
The vast majority of mutant alleles are hidden inside heterozygous carriers, shielded from the pressures of natural selection by the wild-type dominant allele.
Now, Hardy-Weinberg math treats every allele as fundamentally equal. An allele inherited from the mother is mathematically and functionally identical to an allele inherited from the father.
But biology rarely respects clean mathematics.
Right. Sometimes the parent of origin dictates entirely whether a gene functions or catastrophically fails.
This brings us to epigenetics and genomic imprinting.
Mendelian genetics assumes that having 2 copies of a gene, one maternal, one paternal, means both are actively transcribed.
But genomic imprinting introduces an epigenetic override. For certain specific loci, one allele is heavily methylated and actively silenced based solely on its parent of origin.
Only the allele from the other parent is transcriptionally active.
We need to define methylation at the molecular level. We aren't altering the underlying A, C, T, G sequence of the DNA.
We're adding a methyl group, right? A chemical tag directly to the cytosine residues, often at promoter regions.
Exactly. It acts as a permanent molecular mute button. It tightly coils the chromatin, physically preventing the RNA polymerase machinery from accessing and reading the gene.
Because of this deliberate silencing, the individual is functionally haploid for that specific gene. They are entirely reliant on the single unmethylated copy to produce the necessary proteins.
And if that single active copy is mutated or deleted, the cell has absolutely no backup system. The silenced copy cannot be unmuted to compensate.
Imagine you inherit 2 identical steel safes, one from your mother and one from your father. Both safes contain the exact same instruction manuals for neurological development.
Okay, I like this analogy.
But genomic imprinting means the combination to one of those safes has been permanently scrambled. You only have access to the instructions in the other safe.
Right.
If those accessible instructions have pages ripped out, the cell is entirely helpless, even though a perfect copy of the manual is sitting right next to it in the locked safe.
That is a perfect way to visualize it. The clinical consequences of this mechanism are devastatingly illustrated by 2 distinct syndromes mapped to chromosome 15, Prader-Willi syndrome and Angelman syndrome.
Both involve the exact same chromosomal region, 15q11 to q13, but the clinical phenotype diverges entirely based on the parent of origin.
Let's examine Prader-Willi syndrome first. In this specific region of chromosome 15, the maternally derived genes are naturally imprinted. They are methylated and silenced.
So the developing fetus relies entirely on the paternally derived alleles for normal function.
So if a microdeletion occurs on the paternal chromosome 15, removing those active genes, the patient has 0 functional copies. The maternal genes are physically present but transcriptionally dead.
Furthermore, about 25% of Prader-Willi cases are actually caused by maternal uniparental disomy. We just discussed UPD.
The child inherits 2 copies of chromosome 15 from the mother, Both of those copies undergo normal maternal imprinting and are silenced. Again, 0 functional copies.
The pathophysiology of Prader-Willi is deeply tied to hypothalamic dysfunction.
Clinically, it presents with profound neonatal hypotonia and feeding difficulties initially, which paradoxically transitions into severe, insatiable hyperphagia.
The patient lacks the neurological satiety signals to stop eating, leading to rapid life-threatening obesity. It also includes hypogonadism and intellectual disability.
The mnemonic here is POP, Prader-Willi, obesity, paternal allele deleted.
Now let's look at the inverse, Angelman syndrome. Within that same 15q11 to q13 region, there is a specific gene called UBE3A,
which encodes a ubiquitin lysis enzyme critical for protein degradation in the brain.
For the UBE3A gene, the imprinting pattern is reversed. The paternally derived UBE3A is methylated and silenced. The brain relies exclusively on the maternally derived UB3AA.
So if a maternal allele is deleted or mutated, or if the child inherits 2 copies of the paternal chromosome through paternal UPD, the brain has no functional UB3A.
Going back to the analogy, the maternal safe is broken and the paternal space is locked.
Exactly. Without that ubiquitin ligase, specific proteins accumulate toxically in the neurons, profoundly disrupting synaptic function.
The clinical presentation of Angelman syndrome is starkly different from Prader-Willi. It involves severe intellectual disability, microcephaly, frequent seizures, an ataxic puppet-like gait, and characteristic paroxysms of inappropriate laughter.
The mnemonics for Angelman are MAMAS: maternal allele deleted, Angelman syndrome, mood, ataxia, seizures. Or another common one is hails the angels: hand flapping, ataxia, intellectual disability, laughter, seizures.
It is a stunning reality that the DNA deletion can be physically identical, located in the exact same neighborhood of the chromosome, but the parent who handed you that chromosome completely dictates the nature of the neurological collapse.
It really underscores that the sequence itself is not the entire code. The epigenetic regulation, the instructions on how to read the code, is equally vital to human development.
As we transition from the highly specific epigenetic silencing of imprinting back to more standard molecular failures, let's look at the classic modes of inheritance and execute a massive deep dive into one of those rigorously studied diseases in genetics, cystic fibrosis.
To contextualize CF, we must quickly baseline the standard inheritance patterns. Autosomal dominant disorders typically involve defects in key structural proteins or receptors.
They exhibit vertical transmission appearing in multiple successive generations.
They often display pleiotropy, where a single mutation affects multiple seemingly unrelated organ systems, and variable expressivity, where the severity differs even within the same family.
Now, autosomal recessive disorders, like CF, are functionally entirely different. They usually involve deficiencies in enzymes or crucial transport channels.
Because biological systems usually produce excess enzymes, a single healthy allele is generally sufficient. You need 2 defective alleles to manifest the disease.
Therefore, they often skip generations, appearing horizontally in a single SIB-ship, and present with much more severe early-onset phenotypes. The risk increases exponentially with consanguinity.
We also have X-linked recessive disorders, which primarily affect males due to homozygosity. They skip generations through female carriers and feature absolutely 0 male-to-male transmission.
Fathers pass their Y chromosome to their sons, ensuring the defective X goes only to their daughters, making them carriers.
Though it's worth noting that females with Turner syndrome, who are 45 XO and only possess a single X chromosome, are just as vulnerable to X-linked recessive disorders as males.
Finally, X-linked dominant disorders, which are quite rare, are notable because an affected father will transmit the disease to 100% of his daughters, but 0% of his sons.
Let us apply these principles to the ultimate autosomal recessive pathology, cystic fibrosis. The genetics are precise. It is an autosomal recessive defect in the CFTR gene located on chromosome 7.
The most prevalent mutation, responsible for the vast majority of cases in populations of European descent, is a 3-base pair deletion that removes the single amino acid phenylalanine, position 508, hence the delta F508 mutation.
I want to meticulously trace the cellular chaos that this single missing amino acid unleashes because it is a masterclass in pathophysiology.
Let's do it.
The CFTR gene encodes a massive, complex transmembrane protein. It is an ATP-gated chloride channel. In healthy epithelial cells lining the respiratory and gastrointestinal tracts, this channel sits on the apical membrane, facing the lumen, and actively secretes chloride ions out of the cell.
But the delta F508 deletion does not destroy the protein's ability to function as a channel, right?
Right. The devastating problem is structural. The missing phenylalanine causes the CFTR protein to slightly misfold in the endoplasmic reticulum.
And the cell's internal quality control mechanism, the proteasome system, recognizes this misfolded conformation as defective.
So instead of packaging the protein into a vesicle and shipping it to the cell membrane, the proteasome system actively targets it for degradation.
The protein is destroyed before it ever reaches the surface.
Exactly. The apical membrane is left utterly devoid of CFTR channels. This halts chloride secretion into the lumen.
But the pathophysiology doesn't stop there.
No, it doesn't. CFTR physically interacts with and strongly inhibits another channel called ENES, the epithelial sodium channel, which constantly reabsorbs sodium from the lumen back into the cell.
So because CFTR is absent, the inhibitory brake is completely removed from ENAS.
ENES goes into overdrive, massively increasing the reabsorption of sodium ions from the airway lumen back into the intracellular space. And the fundamental law of cellular physiology dictates that water osmotically follows sodium.
The hyperactive sodium reabsorption aggressively pulls water out of the airway lumen and back into the tissue.
The mucus that is normally secreted to line the respiratory tract is suddenly robbed of its hydration. It becomes hyperviscous, incredibly thick, and physically immovable by the mucociliary escalator.
This dehydrated, stagnant mucus plugs the small airways. It creates a perfect hypoxic breeding ground for highly destructive bacterial pathogens.
Infants and young children are plagued by recurrent Staphylococcus aureus infections. As patients reach adulthood, Pseudomonas aeruginosa, often forming impenetrable biofilms, becomes the dominant tissue-destroying pathogen.
The chronic inflammation leads to bronchiectasis, permanent dilation and scarring of the airways, visible as a reticulonodular pattern on a radiograph.
And the dehydration of secretions causes multi-organ collapse. In the pancreas, the thick secretions physically obstruct the pancreatic ducts.
The digestive enzymes trapped inside begin to auto-digest the pancreas itself, leading to severe exocrine pancreatic insufficiency.
Patients cannot absorb fats or proteins. They suffer from severe steatorrhea and become profoundly deficient in fat-soluble vitamins A, D, E, and K, leading to coagulopathies and osteopenia.
The progressive damage to the pancreatic islet cells eventually causes cystic fibrosis-related diabetes. In the liver, the biliary ducts can plug, leading to focal biliary cirrhosis.
It even impacts early neonatal life, presenting as meconium ileus, where the infant's first stool is so thick it causes a complete bowel obstruction.
And the reproductive consequences are nearly universal. Males are almost universally infertile due to the congenital bilateral absence of the vas deferens. The tubes simply fail to develop properly in utero.
Females face subfertility due to highly viscous cervical mucus blocking sperm motility.
The diagnosis of CF often relies on an incredibly unique physiological quirk. While CFTR secretes chloride in the lungs and pancreas, its function is entirely reversed in the sweat glands.
In the acrine sweat ducts, CFTR is supposed to reabsorb chloride from the sweat before it reaches the skin surface.
Because the channel is missing, the chloride stays trapped in the sweat. This creates the classic elevated chloride concentration on a pilocarpine-induced sweat test.
The sweat is pathologically salty, which can lead to severe hypochloremic, hypokalemic contraction alkalosis if the patient sweats excessively.
For decades, the management of this multi-system failure was purely reactive. We deployed aggressive chest physiotherapy to physically percuss and dislodge the concrete-like mucus.
We use inhaled Dornase alpha, a recombinant DNase enzyme that literally chops up the highly viscous DNA released from dead neutrophils in the airway to thin the sputum. We prescribed massive doses of pancreatic enzyme replacements with every meal.
But the narrative of CF has radically shifted in the last decade due to the development of CFTR modulators. This is the absolute bleeding edge of pharmacogenomics.
We are no longer just fighting the downstream symptoms. We are physically interacting with the mutated protein itself.
The modulators are categorized by their specific mechanism of action.
The first class are the potentiators, like ivacaftor. These are designed for patients who have specific mutations where the CFTR protein actually reaches the cell membrane, but the gate is structurally stuck in the closed position.
The potentiator physically binds to the defective channel and forces the gate open, restoring chloride flow.
But what about the majority of patients with the delta F508 mutation, where the protein misfolds and is destroyed before it ever reaches the surface? A potentiator is useless if the channel isn't there to open.
It requires the second class of modulators, correctors, such as lumicaftor or tizacaftor.
These molecules act as pharmacological chaperones. They enter the endoplasmic reticulum, bind to the misfolded delta F508 CFTR protein and artificially stabilize its three-dimensional conformation.
This shields it from the cell's quality control proteasomes, allowing the protein to traffic successfully to the cell membrane.
And once the correctors deliver it to the membrane, they use a potentiator to help it function optimally.
It is molecular engineering at its most precise. We are deploying bespoke chemical chaperones to fix a folding error caused by a single missing amino acid.
From the misfolding of ion channels, we must shift our focus to mutations that destroy the very physical architecture of our cells. This brings us to the muscular dystrophies and several devastating rare syndromes.
While CF is the quintessential autosomal recessive disease, Duchenne and Becker muscular dystrophies are the textbook examples of X-linked recessive inheritance.
The pathology centers entirely on the DMD gene, which encodes the protein dystrophin.
The dystrophin gene holds a distinct record in human biology. It is the largest protein-coding gene in the entire human genome, spanning roughly 2.4 megabases.
Because the gene is physically so massive, it is an exceptionally large target. It has an extraordinarily high rate of spontaneous de novo mutations simply because there is so much DNA to copy during replication.
What exactly is dystrophin doing that makes its loss so catastrophic for muscle tissue?
Dystrophin serves as a critical mechanical shock absorber. It acts as an intracellular anchor, connecting the internal actin cytoskeleton of the muscle fiber to the transmembrane dystroglycan complex, which in turn anchors to the extracellular matrix.
Every time a muscle contracts, massive shearing forces are generated across the cell membrane.
Dystrophin distributes that force safely. Without it, the mechanical stress of simple, everyday muscle contraction literally tears microscopic holes in the sarcolemma, the muscle cell membrane.
The tears allow massive influxes of calcium into the cell, which activates destructive proteases, leading to relentless myonecrosis, the physical death of the muscle fibers.
In Duchenne muscular dystrophy, the mutation is typically a frameshift deletion or a nonsense point mutation.
Meaning, the mutation alters the downstream reading frame of the mRNA sequence, introducing a premature stop codon. The ribosome halts translation early, resulting in a severely truncated, entirely non-functional protein that is rapidly degraded.
The dystrophin is functionally absent. Because the anchor is gone, the destruction is rapid and progressive. Clinically, Duchenne presents in early childhood, typically before age 5.
Weakness begins in the proximal muscles of the pelvic girdle and progresses superiorly. A highly distinctive clinical sign is pseudohypertrophy of the calves.
The calf muscles appear massively enlarged, but it is an illusion. The muscle tissue itself has died and is being progressively replaced by structurally useless fibrofatty connective tissue.
The weakness forces patients into a waddling gait. It also produces Gower's sign, a specific clinical maneuver where a child, attempting to stand from a seated position on the floor, must physically walk their hands up their own shins and thighs to brace their collapsing pelvic muscles.
It is a devastating progression, and because dystrophin is also expressed in the myocardium, dilated cardiomyopathy is inevitable, frequently becoming the ultimate cause of mortality in young adulthood.
Now contrast that rapid destruction with Becker muscular dystrophy.
Becker is caused by mutations in the exact same DMD gene, but the nature of the mutation is fundamentally different. Becker is typically caused by non-frameshift deletions.
A chunk of the gene is missing, but the remaining sequence is still in the correct reading frame.
The cell produces a dystrophin protein that is internally shortened but still retains its essential anchoring domains at either end. It is partially functional.
The rhyming mnemonic here is Becker is better. Because the mechanical anchor partially works, the onset of symptoms is delayed until adolescence or early adulthood, and the clinical course is far less severe.
The distinction between a frameshift and a non-frameshift mutation dictates the entire trajectory of the patient's survival. Shifting gears entirely, let's look at a structural muscular disorder driven by an entirely different genetic mechanism: myotonic dystrophy type 1.
This is an autosomal dominant disorder caused by an abnormal expansion of a CTG trinucleotide repeat in the DMPK gene. We are going to explore the mechanics of trinucleotide repeats extensively in our final section, but the pathophysiology here is deeply unique. It involves RNA toxicity.
The massive expansion of CTG repeats creates heavily mutated mRNA transcripts. These toxic transcripts remain trapped in the nucleus and physically sequester critical splicing proteins, particularly muscle-blind-like 1 or MBNL1.
Because MBNL1 is trapped, it cannot properly splice the mRNA for various downstream muscle proteins, including a critical muscle-specific chloride channel, CLC1.
Without that chloride channel functioning properly, the muscle cell membrane becomes hyperexcitable. This leads to the hallmark clinical sign, myotonia, the delayed relaxation of skeletal muscle after voluntary contraction.
If a patient grips your hand tightly or grasps a doorknob, the electrical instability prevents the muscle from releasing. They physically cannot let go.
The clinical presentation is broad. The text mnemonic for myotonic dystrophy relies on the repeat sequence itself, CTG. Cataracts, toupee referring to early frontal balding in males, and gonadal atrophy. A classic muscle biopsy will show highly abnormal ring fibers and centrally located nuclei confirming the structural disarray.
From the structural architecture of the cell, we must look at the cellular power grids. The text transitions into rare mitochondrial and sporadic diseases. We previously discussed how mitochondrial DNA is strictly maternal and subject to heteroplasmy.
Because the mitochondria's sole job is oxidative phosphorylation, mutations here selectively devastate tissues with massive metabolic demands, the brain and skeletal muscle.
This manifests as mitochondrial myopathies, such as Mellis mitochondrial encephalomyopathy, lactic acidosis and stroke-like episodes, and Murph myoclonic epilepsy with ragged red fibers.
The histology is striking. When a muscle biopsy is stained with Gamori trichrome, you see the classic ragged red fibers.
That structural abnormality is actually a desperate physiological response. Because the mutated mitochondria are failing to produce sufficient ATP, the muscle cell senses starvation.
It attempts to compensate by furiously replicating the defective mitochondria, creating massive aggregates of dysfunctional organelles that cluster under the sarcolemma, appearing as jagged red borders under the microscope.
Another severe manifestation of mitochondrial failure is Leber hereditary optic neuropathy. This is typically driven by point mutations in the mtDNA genes encoding complex phi of the electron transport chain.
The highly metabolically active retinal ganglion cells and their axons in the optic nerve undergo catastrophic apoptosis. It presents in teens or young adults as subacute, painless, but permanent bilateral vision loss.
The final syndrome we need to analyze in this section operates on yet another distinct mechanism, Rett syndrome. Unlike the inherited diseases we've discussed, Rett syndrome is almost universally a sporadic de novo mutation. It involves the MECP2 gene, located on the X chromosome.
The MECP2 protein is a transcriptional repressor. It binds to methylated DNA and shuts down the expression of various target genes, orchestrating the precise timing of synaptic maturation in the developing brain. If MECP2 is mutated, that regulation is lost.
Because it is X-linked and functionally so crucial, a MECP2 mutation is generally lethal in homozygous male embryos. They do not survive. Therefore, Rett syndrome is seen almost exclusively in heterozygous females who survive due to X inactivation, providing some functional cells.
The clinical timeline of Rett syndrome is exceptionally cruel. For the first 6 to 18 months of life, the infant's development appears completely normal. They hit their milestones, they learn to walk, they acquire rudimentary language.
And then, the MECP2 failure reaches a critical threshold in the maturing brain. The text uses a poignant spelling mnemonic for the regression. Return, spelled R-E-T-T-U-R-N.
The child undergoes a severe, rapid loss of all previously acquired motor, cognitive, and verbal skills. They develop ataxia, growth deceleration, seizures, and severe autonomic dysfunction.
The most pathognomonic sign is the replacement of purposeful hand use with relentless, stereotypic hand-wringing or hand-washing movements.
It is a devastating demonstration of how the delayed failure of a single epigenetic regulator can utterly dismantle an initially normal neurodevelopmental trajectory.
And this brings us to the most insidious mechanism of inheritance we will cover today. We have examined alleles that misfold, alleles that are silenced, and alleles that tear cells apart. But we must now examine a mechanism where the genetic typo physically multiplies itself, growing larger and more destructive as it passes from parent to child.
This is the phenomenon of genetic anticipation, driven by trinucleotide repeat expansions. The DNA sequence develops a literal stutter.
Let's drill down into the mechanics of this stutter. Imagine a localized region of a gene that contains a sequence of 3 nucleotides repeated over and over, such as CGG, CGG, CGG.
During DNA replication or repair, the DNA polymerase machinery has to copy this highly repetitive track. Because the sequence is so uniform, the newly synthesized strand can temporarily detach and misalign, forming a physical hairpin loop.
The polymerase essentially loses its place. When it resumes copying, it accidentally synthesizes extra copies of the triplet. What was a stable sequence of 30 repeats in a parent can suddenly expand into an unstable sequence of 80 repeats in the gamete.
With each successive generation, the track grows longer, the physical instability increases, and the clinical impact intensifies.
That intensifying clinical impact is the definition of genetic anticipation. The disease severity progressively increases, and the age of onset progressively decreases in each successive generation of a family.
The quintessential model of this mechanism is Fragile X syndrome. It exhibits an atypical, highly complex X-linked dominant inheritance pattern. The mutation is a massive expansion of CGG repeats located in the 5' untranslated region of the FMR1 gene on the X chromosome.
The FMR1 gene encodes a critical RNA-binding protein that shuttles mRNA to the dendritic spines of neurons, governing synaptic plasticity. A massive CGG expansion in the promoter region triggers intense hypermethylation.
The cell's epigenetic machinery recognizes the massive abnormal repeat as a threat, slaps methyl groups all over it, and permanently silences the entire FMR1 gene.
The timing of this expansion is deeply sex-specific. The massive leap from a moderate premutation to a full disease-causing mutation occurs almost exclusively during maternal oogenesis, the creation of the egg cells.
The mnemonic here is fragile EGG, linking the CGG repeat to oogenesis. The clinical impact of losing the FMR1 protein is profound. Fragile X is the most common inherited cause of intellectual disability.
While Down syndrome is the most common genetic cause overall, the vast majority of trisomy 21 cases are sporadic non-disjunction events, not inherited familial traits.
The presentation of Fragile X is entirely dependent on the exact length of the trinucleotide stutter. Individuals with a premutation carrying between 50 and 200 CGG repeats do not have the classic syndrome because the gene is not yet fully silenced.
However, the massive toxic mRNA transcripts produced by the expanded sequence can cause Fragile X-associated tremor and ataxia syndrome in older males, or primary ovarian insufficiency in females.
But if the sequence expands beyond 200 repeats during oogenesis, creating a full mutation, the hypermethylation completely shuts the gene down. These patients present with the classic Fragile X phenotype.
Physically, this includes postpubertal macroorchidism, significantly enlarged tests, a long, narrow face with a prominent jaw, large averted ears, and hypermobile joints.
Neurologically, it presents with moderate to severe intellectual disability and significant autistic spectrum behaviors, including poor eye contact, social anxiety, and repetitive actions. The clinical literature also highlights that severe self-mutilation behaviors can occur, requiring careful diagnostic differentiation from Lesch-Nyhan syndrome, another disorder characterized by self-injury due to hyperuricemia.
To round out our understanding of genetic anticipation, we have to acknowledge the other major trinucleotide repeat disorders. While we won't dissect their full pathophysiology today, they follow the exact same mechanism of a slipping polymerase causing progressive intergenerational destruction.
We have Huntington disease, an autosomal dominant neurodegenerative disorder. The repeat is CAG, occurring within the coding region of the HTT gene, leading to a toxic polyglutamine tract that destroys neurons in the striatum.
The mnemonic is caudate as decreased ACE and GABA, tying the CAG repeat to the specific neurotransmitter loss in the caudate nucleus.
We previously analyzed myotonic dystrophy with its CTG repeat in the DMPK gene causing RNA toxicity and splicing errors, autosomal dominant with the mnemonic CTG covering cataracts, toupee, and gonadal atrophy.
Then Fragile X, the CGG repeat, X-linked dominant with the visual mnemonic chin protruding, giant gonads matching the CGG sequence.
And finally, Friedreich ataxia. This is an autosomal recessive disorder caused by a GAA repeat expansion in the frataxin gene, leading to severe mitochondrial iron accumulation and spinocerebellar degeneration.
The mnemonic is straightforward. Ataxic GAA, explicitly linking the broad-based staggering gait to the GAA repeat.
Ataxic GAA. The sheer volume of highly specific genetic data requires these mental scaffolding techniques.
As we bring this intensive deep dive to a close, let's look back at the incredible architecture we've deconstructed today. We began by analyzing the sabotage of dominant negative mutations, where a single broken p53 subunit poisons an entire tetramer.
And the life-saving localized glitch of somatic mosaicism in McCune-Albright syndrome.
We then utilized the null hypothesis of the Hardy-Weinberg equilibrium to prove that an allele can be shockingly pervasive, like the 3.4% carrier frequency for cystic fibrosis, silently flowing through the population gene pool, hidden within asymptomatic heterozygotes.
We unlocked the epigenetic vaults of genomic imprinting, recognizing that the severe hyperphagia of Prader-Willi or the ataxic seizures of Angelman syndrome are dictated dictated entirely by the parent of origin methylation of chromosome 15.
We mapped the exact cellular plumbing failure caused by the misfolded delta F508 protein in cystic fibrosis and marveled at the correctors and potentiators engineered to physically reshape it.
We traced the mechanical collapse of the sarcolemma in Duchenne muscular dystrophy back to a frame shift deletion in the massive dystrophin gene.
And we ended by watching the genetic code physically stutter and expand across generations, creating the intensifying neurological collapse of trinucleotide repeat disorders like Fragile X.
Grasping these complex molecular pathways, understanding exactly how a gene is silenced by a methyl group, destroyed by a proteasome, or expanded by a slipping polymerase, it is about far more than just categorizing pathology.
It absolutely is.
It is about deeply understanding the delicate statistical miracle that is every single healthy human body. Every single step in the biochemical cascades we discussed today presents a massive opportunity for catastrophic failure.
And yet the vast majority of the time, the cellular machinery repairs the strand, the protein folds correctly, and the code holds.
It is a profound testament to the redundancy and resilience of human biology.
It is a statistical miracle playing out in every cell. But I want to leave you with a final provocative thought regarding the evolutionary implications of the mechanisms we just explored.
Let's hear it.
If the phenomenon of genetic anticipation guarantees that trinucleotide repeat diseases like Fragile X or Huntington's will inherently get progressively worse, expanding and striking earlier with every single successive generation, what does this imply for the long-term evolutionary future of those specific alleles?
That is the ultimate consumption of population genetics. If the age of onset continually decreases generation after generation until the disease strikes before the individual reaches reproductive age, will the mechanics of natural selection eventually force these specific stuttering alleles to eradicate themselves entirely from the human gene pool?
Or is the architecture of human DNA forever destined to randomly generate and carry these molecular echoes, constantly stuttering, expanding, and silencing in an endless, chaotic dance? It's something to mull over. Thank you for joining us in the diagnostic deep waters today. Stay insanely curious, keep questioning the blueprints, and we will see you next time.